Hello, 8th standard CBSE children. Have a good day. Have a nice day. Today we are going to continue the lesson chapter 2 microorganisms friend and foe as video number 20. Right? We are doing video number 20. In previous three videos, that is 17, 18, 19, we have learned in detail about the microorganisms as what are microorganisms, friendna, their useful activities, and fauna, their harmful activities. Then continuation of that lesson in our textbook is about food preservation. There are various methods of food preservation. I have given in detail about food preservation, referring various possibilities, because in our textbook is given very little with the picture presentation we have done in previous video as food preservation. Video number 19 la food preservation பத்தி படிச்சிருக்கோம். Now in this video number 20 we are going to do the summing up of this microorganism. Still I have better pictures and figures as we call that. We will continue this video number 20 and complete the lesson chapter 2 microorganisms friend and foe. I hope I am clear, right? So microbes are found almost everywhere on earth. They can be classified broadly as bacteria, algae, fungi, protozoa and viruses. Yeah, this is namakku introduction. சரியா? Now this introduction again, as we say, microbiology means study of microbes. Occurrence, wherever it is present, that we name it as occurrence. It is present in ponds, lakes, river, ocean and soil. I repeat, they are occurring in ponds, lakes, rivers, ocean and soil. And it can be in the adverse condition. Adverse condition, very unfavorable condition, as hot spring, desert, deep oceans. Hot spring na nalla chooda irukku adu, and desert na very hot climate, and deep oceans na kadal kadiyila kuda these microbes can occur. So that is occurrence of these microbes even during adverse condition as hot spring, desert, deep oceans. Right? Now, what are the categories of microbes? Otherwise, we say infectious agents of microbes. They are viruses, bacteria, fungi, algae, and protozoa. I repeat, viruses, bacteria, fungi, algae, and protozoa. சரியா? So எல்லாம் சொல்லிட்டேன்னு நினைக்கிறேன். Viruses, bacteria, fungi, algae and protozoa. Okay. Now next one, what are viruses? Viruses, it means poison in Latin, and the study of virus is called virology. And viruses are 10,000 times smaller than bacteria, and it is made up of only genetic material and protein. Other detailed cell structure is absent in viruses. So they are made up of, viruses are made up of genetic material and protein, and it is said to be intracellular obligatory parasite. What does it mean? It can live only as parasite. It is as intracellular, within the living tissues only it can survive. So it is as intracellular obligate parasite, and actually viruses are said to be intermediate between living and non-living things. I repeat, viruses are intermediate form between living and non-living things. Okay, and there are different shapes as rod shape, spherical, cylindrical, and tadpole like, not particular, tadpole like கூட இருக்கும். So there are different shapes and sizes of viruses, right? So viruses can be present as living cell only within the living host, and the viruses are considered to be non-living, which can be crystallized and not capable of any life, that is as non-living character. I hope you understand. Yeah? This is for virus. Now the structure of virus, as we know, the genetic material is made up of DNA or RNA, any one only. Genetic material is made up of DNA or RNA. And protein coat covered, it has a protein coat, outer protein coat covered by an envelope that is called capsomere. At the figure that is called capsomere. And this envelope is as protein, lipids and carbohydrates. And the envelope has spike. Spike na குச்சி குச்சியா இருக்கும்ல, இப்ப நம்ம கொரோனா வைரஸ் கூட பார்க்கிறோம்ல, அதுல நிறைய அப்படி துருத்தின் இருக்கும்ல, அதுதான் ஸ்பைக். I hope you understand. So structure of virus, it has genetic material as DNA or RNA, and protein coat is covered by an envelope, and that envelope has spike. Spike na enna sonne, area projections like coronavirus. Coronavirus nariya paathirukilla, right? Now idla paru, this is a influenza virus, spherical. In this picture, it is an influenza virus causing the fever influenza. சரியா? And the spike தெரியல, surface protein இருப்பாரு, அதுதான் spike, right? And the outer protein coat, it is known as capsid. Outer protein coat is known as capsid. சரியா? And the genetic material here, it is RNA. What is RNA? Ribonucleic acid. சரியா? The genetic material is RNA, ribonucleic acid. DNA na deoxyribonucleic acid. I hope you understand. Yeah? So what is the structure of virus for influenza? It is a spherical virus, and then it has a outer coating called surface protein with the spikes, and then the brown cover theriyal, adu vandu capsid, the protein coat, and the genetic material here, it is RNA. RNA expansion, what I said, it is ribonucleic acid. I hope you understand. The next one, the living characters of virus. Living characters of virus as respond to heat, chemical and radiation. Viruses will respond to heat, chemicals and radiation, and it can reproduce only inside the host cell. Viruses can reproduce only inside the host cell. And then it shows irritability. Irritability in a response to stimuli. When viruses are triggered, it shows irritability. Respond to stimuli. Then non-living, these three are living characters. And one more you can add, it is capable of having life activities when it is present within the living host. And non-living character, it is inactive when present freely in the environment. Non-living character, when present freely in the environment, it is inactive, and it can be crystallized, and it has no metabolic machinery. Synthesis of protein or nucleic acid and cytoplasm, they are absent. I hope you are listening. So this is for living and non-living characters of viruses. Sorry? The next one, different shapes of virus. Super padam idellame. Different shapes of virus. First one, tobacco mosaic virus. Second, influenza. Second, influenza. And third one, it is bacteriophage. The reverse like A, B, C. A bacteriophage, it is a tadpole-like structure. Bacteriophage, phage in a virus. Virus, when it is affecting the other host, it is called phage. When it is attacking the bacterium, it is called bacteriophage. When it attacks any other plant, it is phytophage. When it is doing any animal, it is zoophage. And ALGAE na phycophage and fungus na microphage. So phage referring to virus. And then middle one we have seen B influenza, spherical shape with the surface protein, capsid and RNA. Whereas tadpole and the head with the capsid and the protein, and within that DNA, deoxyribonucleic acid, genetic material is present. And third one, C, tobacco mosaic virus. That is the virus when it is attacking the tobacco plant, it is called tobacco mosaic virus. And the brown they are called capsomeres. They are the outer protein coat called capsomeres. And then the green spiral, that is the genetic material RNA. I hope you are listening. So here, understand the genetic material, it is as either DNA and RNA, but never both. So tobacco mosaic genetic material RNA, influenza virus RNA, whereas bacteriophage it is DNA. I hope you understand. Can you? The next one, bacteria. Bacteria, they are bacterium singular, bacteria plural. They are single celled prokaryotes. What are prokaryotes? நம்ம cell structure la படிச்சிருக்கோம்ல prokaryotes. They are unicellular organisms with the nuclear material represented only by the genetic material, absence of nuclear membrane and nucleolus. And normally it is unicellular and there is no well-defined nucleus and cell organelles are present. Cell organelles are absent. So they are prokaryotic structure. And the bacteria are said to be first living organisms on earth. And it comes under the classification kingdom Monera. Actually, this kingdom as Whittaker, the taxonomist, classified all living organisms as 5 kingdoms. So it is as 5 kingdom classification as kingdom Monera, Protista, Fungi, Plantae and Animalia. 5 kingdom classification on the kingdom Monera, kingdom Protista, kingdom Fungi, kingdom Plantae and kingdom Animalia. Adula first occurrence on the kingdom Monera. And then the study of bacteria is called bacteriology. Normally, the size of the bacterium is 1 micrometer to 5 micrometer. I hope you understand. And then the bacteria can be classified based on different criteria. One is based on respiration. It is as aerobic and anaerobic bacteria. And then based on the presence of Flagellum, the locomotory organ, bacteria can be classified as monotrichous, lophotrichous, amphitrichous, peritrichous, and atrichous. I hope you are listening. Based on the presence of flagella, it is as monotrichous, one flagellum, lophotrichous, both the side tuft of flagella, amphitrichous, one side tuft of flagella, and then the presence on the diagram which I will show you, and then amphitrichous, peritrichous, and atrichous bacterium. And then bacteria can be classified based on different size and shape, and bacteria can be classified based on the response to gram strain. So that is for classification. Now bacteria cell structure, it has outer covering cell wall, nuclear material, it is called nucleoid or incipient nucleus, that is without nucleolus and nuclear membrane. It has nucleolus and nuclear membrane. Right? There is another extra DNA material called plasmid is present, which is characteristic of this bacterial cell structure. And the locomotory organ, it is flagella. Right? And cell organelles are absent. Another mitochondria, Golgi body, lysosome, chloroplast, are absent. But bacteria can do photosynthesis by the presence of bacterial chlorophyll. It has no green chlorophyll, but it can do photosynthesis by bacterial chlorophyll. Now see the structure. The nucleoid, it is circular DNA, where it is called incipient nucleus. So here the cell structure, what you are seeing, cell wall, cell membrane, outer covering is called capsule, and then it is covered by flagella. But there are one or two flagella structure that is called pili, which is helpful in reproduction. And there is another extra plasmid on the cytoplasm between the cell membrane labeling of cytoplasm, in between plasmid will be present. Labeling I hope you understand. The next one, classification. When the bacterium, it is rod shape, it is bacillus. Spiral shape, spirillum. Cocci, it is round shape. Vibrio, it is comma shape. Bacillus, bacillus singular, bacilli plural. Spirillum singular, spirilla plural. Coccus singular, cocci plural. So bacillus rod shape, spirilla spiral shape, coccus spherical, and vibrio comma shape. Right? Now see the figure: bacillus rod shape, spherical, comma, and spiral. I hope you understand. The vibrio oru flagellum that is called monotrichous. And tuft of flagella at one side, it is lophotrichous. The lophotrichous, amphitrichous வந்து நான் சொல்றேன், அப்ப புரிஞ்சுக்கோ. ஒரு இது ஒரு சைடு tuft of flagella, இன்னொன்று both side tuft of flagella. The வருது பாரு. So number and arrangement of flagella: monotrichous, lophotrichous, amphitrichous, peritrichous, and atrichous. See the level. Super. Monotrichous, one flagellum, amphi, one at one end, both the end. Lopho on the tuft at one end, peritrichous all over the body. Atrichous, no flagellum. I hope you follow. Further diagram for number and arrangement of flagella. Types தெரிஞ்சுக்கோ. இதெல்லாம் additional points நான் கேட்பேன். சிலது நம்ம bookல இல்லை, ஆனா நான் கேட்பேன். சரியா? Then fungus, fungi, it is a eukaryotic organism. It lacks chlorophyll, absence of chlorophyll. Then it is unicellular, example yeast. Multicellular, example penicillium. and macroscopic mushroom. Mushroom. And study of study of fungus is called mycology. And then it comes under 5 kingdom classification, kingdom fungi. I hope you understand. The next one, yeast. Unicellular fungus is called yeast. It grows in it grows in all kind of media with the sugar, and it is oval in shape. It has cell wall, nucleus, glycogen, and respiration is by anaerobic method, and yeast reproduce by budding. See the diagram. Outer cell membrane, presence of vacuole cytoplasm, and then large nucleus is present. And yeast reproduces by budding. I hope you understand. So this is for yeast. Next one, mushroom. Normally it is found during rainy season. ஒரு சின்ன மழை பெஞ்சா போன இந்த மஷ்ரூம் நாய்க்கூட முளைக்கும்ல அதான் rainy season. Umbrella shape. The fruiting body is a umbrella shape structure called basidiocarp, and then within that it has fine lamellar structure called gills, which is reproducing by spores. And then the plant body is called mycelium, which in turn hyphae, which in turn called mycelium, where the cell wall material is chitin and cellulose. And fungi reproduces by fragmentation and spore formation, and nutrition characterized by saprophytic, parasitic, and symbiotic nutrition. Nutrition is characterized by saprophytic, parasitic, and symbiotic nutrition. Now what you are seeing here is a mushroom. Now can you see that dark grey? They are as underground hyphae. This hyphae are filamentous structure formed of mycelium, and one of that hyphae, when it's growing and developing into the fruiting body, the cap netla, that whole structure, the brown structure pere basidiocarp. Adla cap mari irukum. The stalk called pileus. Then keela gills nettu pathiya, they are the structure producing the spores. I hope you understand. இது வந்து நாய்க்கொடை நம்ம தமிழ்ல சொல்லுவோம்ல. Okay. Then algae, they are eukaryotic organism rich in chlorophyll, and it is forming a grass-like water. Algae in the கோவில் குளத்துலலாம் பார்த்தீங்கன்னா அப்படி பாசி பிடிஞ்சிருக்கும்ல, அதான் grass of water, and it is capable of preparing its own food by the presence of chlorophyll. So it is autotrophic in nutrition, and the study of algae is called algology or phycology, and it may be unicellular, example Chlamydomonas, or multicellular Spirocheta, and it can be filamentous or non-filamentous. It can be motile or non-motile. It can be branched or unbranched. It can occur in single or in colony. I hope you understand. And then this algae is characterized by the presence of the plant body called thallus, where there is no differentiation of root, stem, and leaf. It is only a mass of structure. So Chlamydomonas occurring single, unicellular. Next volvox, it occurs in colony. நிறைய dot green இருக்குல்ல, அது ஒன்னொன்னும் volvox. Then Ulothrix, it is unbranched filament. Then Hydrodictyon, it is a branched filament. Then Ulva, it is a macro algae, and Fritschilla, it is also a type of algae. So these are different types of algae examples. The next one, Chlamydomonas. It's about the Chlamydomonas. Just a brief technical. It is unicellular, showing movement, so motile, freshwater form. The shape is oval or pyriform, and it is found in ditches, water tank. Cell wall is made up of cellulose with a nucleus and characterized by cup-shaped chloroplast. And then near the cup-shaped portion, it is having the contractile vacuole, and the cup portion, the shallow portion, nucleus will be present. And then the locomotory organ, it is flagella, and it has photosynthetic pigment, chlorophyll, so it can prepare its own food. And Chlamydomonas reproduce both by sexual and asexual method. See the diagram, Chlamydomonas. First, flagellum. The cell organelle, it is mitochondria, then cytoplasm, cup-shaped chloroplast, blue on the nucleus, and this chloroplast will have reserve food material as starch grain and then pyrenoid. And then Golgi complex are also present. Diagram is very clear. Particle, I may ask you to draw the diagram for the test. Okay. The next one, it is kingdom Protista. We are talking about protozoa. So far we have done virus, bacteria, fungi. Now protozoa, it comes under kingdom Protista, 5 kingdom concept. And then it is protozoology, and the types are ciliates as paramecium. Example, spelling தப்பில்லை பார்த்துக்கோ, PARAMOE, O விட்டிருக்கல spelling பார்த்துக்கோ. Paramecium, flagellates, euglena, pseudopod, amoeba, sporozoan, plasmodium. I repeat, kingdom Protista, so it comes under protozoology. Types are 4 types, classification. Ciliates, example, paramecium, spelling PARAMOECI. Then flagellates, example, euglena, pseudopod, example, amoeba, and sporozoan, plasmodium. See the diagram. Common protozoans. Plasmodium, center வந்து paramecium, கீழே வந்து euglena, green வந்து euglena. I hope you are able to follow. சரியா? இல்லை கரெக்டாக இருப்பார். Flagellate, presence of flagella, pseudopod, presence of pseudopodia, sporozoan, parasite, plasmodium. I hope you are clear. Then amoeba, நிறைய படிச்சிருக்கோம் இல்லையா? It is an unicellular microscopic structure, cell membrane. It is an outer cell membrane with cytoplasm and nucleus. Pseudopodia or false sweet is present, and then contractile vacuole which regulates osmo regulation. Clear? Reproduction is by binary fission and sporulation. During unfavorable condition, the amoeba withdraw its pseudopodia and develop a cyst around it, and within that the content of the protoplasm gets shrunken, and during favorable condition it bursts open by spores, and then everyone will form a new daughter amoeba. This is sporulation. See the diagram, amoeba. நிறைய படிச்சிருக்கோம் இல்லையா? pseudopodia, food vacuole, cytoplasm, nucleus, contractile vacuole. Very clear? The next one, uses of microbes. நம்ம வந்து friend and phone சொன்னோம் இல்லையா? இது வந்து friend. Medicine as antibiotics. Example, streptomycin, penicillium. What you are seeing here, the green வந்து penicillium. It is penicillium. இந்த structure வந்து இப்படி branch மாதிரி இருக்குல்ல, அதிலிருந்து it reproduces by the structure called conidia. சரி? So that structure is known as conidium. So medicine, antibiotics, Example, streptomycin and penicillin. எதுக்கு? Medicine. Right? Then vaccine, it may be MMR, that is measles, mumps and rubella, BCG, Bacillus, calamine and guar, right? And it is as agricultural. In agriculture, it is acting as natural fertilizers because they are as microbes. So these microbes are decomposers. Fungi and bacteria, they are as decomposers. And nitrogen fixation, the rhizobium and acetobacter, they take part in nitrogen fixation. Yeah, other uses of microbe. Go through panel. Vaccine, MMR and BCG. Agriculture, natural fertilizer, microbes as decomposers, and nitrogen fixing bacteria, another rhizobium and acetobacter. See the diagram, how it is fixing the nitrogen. என்னத்துக்கு? Plants cannot take nitrogen directly from the atmosphere. So, with the help of these nitrogen fixing bacteria, they are helpful in fixing the nitrogen for the green plant. So, this associate the phenomenon, it is called symbiosis, where this bacterium is fixing nitrogen for the plant, and that in turn getting shelter from the plant. So this association where both the partners are mutually benefited, that is called, that association is called symbiosis. And these organisms, they are called symbionts. And they are as biofertilizers, bacterial, fungi, algal, aquatic fern and earthworm. Figure konja fade irukku ana purinjukko. Okay, first rhizobium, second acetobacter, third azospirillum, fourth வந்து எனக்கு புரியலை. அடுத்தது வந்து aquatic fern, கீழே இருக்கிறதெல்லாம் aquatic fern, azolla, அது தெரியல, கொஞ்சம் புரியலை, புரிஞ்சுக்கோ. aquatic fern. சரி? So these are examples of biofertilizers. Okay. The next one, it is called biocontrol agents. இதெல்லாம் இப்ப ரொம்ப important. Biotechnologyல இந்த biocontrol agents லாம் ரொம்ப important. சரியா? It is as Bacillus thuringiensis, Trichoderma and baculoviruses. See the example, first one, Bacillus thuringiensis, where it is having a special gene as BT, அதாவது Bacillus thuringiensis gene, when it is inserted into the crop, the pesticide will get affected. So, pest dies when feeding on any part of the plant. So, the crop is infected by European corn borer, அது பேரு, அந்த cornக்கு பேரு. So, அது வந்து activity of this will kill the pest. Another example, Trichoderma and baculoviruses. These are called biocontrol agents. I hope you understand. The next one, it is in industry, they are also useful. Sewage treatment, example, Nitrobacter species, Methanobacterium, production of biogas, anaerobic bacteria, methane, carbon dioxide and hydrogen. These are additional points புரிஞ்சுக்கோ. சரியா? Then production of alcohol and wine fermentation by yeast, and microbes in retting and tanning process. Example, Pseudomonas aerogenisa. இதில் மெயினாக நீ படிக்க வேண்டியது வந்து anaerobic bacterium, then sewage treatment, production of alcohol and wine, where this yeast is playing a major role. Then in daily life, என்ன சொல்லுவாய்? Daily life, it is making of bread. Yeast, example, yeast and chlorella. Preparation of curd and cottage cheese, example, lactobacillus. Lactobacillus நம்ம beginning of the lesson la படிச்சிருக்கோம் இல்லையா? The lactobacillus acting upon milk and converting them into curd, lactobacillus. And in human intestine, this lactobacillus and E. coli, they are taking part in digestion. சரியா? Then come to harmful activities. கொஞ்சம் blurred இருக்குன்னா புரிஞ்சுக்கோ. This is caused by microorganisms in human: tuberculosis, cholera, common cold, rabies, amoebic dysentery, and malaria. I repeat: tuberculosis, cholera, common cold, rabies, amoebic dysentery, and malaria. சரி? And the causative organism, அதான் எனக்கு மெயினா தெரியணும். Causative organism: tuberculosis, Mycobacterium tuberculosis, cholera, Vibrio cholerae, common cold, influenza virus, and then rabies, rhabdoviridae, and amoebic dysentery by AMOEBA histolytica, and malaria caused by the protozoan plasmodium. சரியா? And then mode of transmission தெரியணும், symptoms தெரியணும். படிக்கலை, நீ பார்த்துக்கணும். Symptoms and preventive measures. கொஞ்சம் லைட்டா இருக்கு, ஆனால் படிச்சுக்கணும். பேர் பண்ணிக்கோ, இன்கன்வீனியன்ஸ் பேர் பண்ணி கொஞ்சம். எனக்கு தள்ளி நின்னா படிக்க முடியாது. I hope you can read better. இல்லையா? Can you? Shall I go to next? ஒரு தடவை go through பண்ணு. Can you? The next one, this is caused by microbes in animals and plants. என்ன சொல்றது? Animals வந்து anthrax, cattle also affects human. Sorry, anthrax, a cattle disease. It also affecting the human. Then foot and mouth disease. Anthrax is caused by Bacillus anthracis. சரியா? And then aptovirus, foot and mouth disease. Mode of transmission பாரு, through contaminated soil and food. இது வந்து air and animal vectors. Right? The anthrax வந்து Bacillus anthracis. Actually இதுக்கு ஒரு சின்ன information சொல்ல. அதாவது this anthrax spores, they are highly poisonous. அதனால in the World War time, especially Second World War, those fighters, they were using this anthrax spores dipped in their arrow. அம்பு விடுவால இந்த arrowல இந்த anthrax spores dip பண்ணி they were fighting, causing so many death. டெத்துக்கு நம்பரே தெரியாம it caused many death. அவ்வளவு highly poisonous. சரியா? Then by microorganism in plant, it is citrus canker, Xanthomonas citri. வேற பேருக்கான புரிஞ்சுக்கோ, Xanthomonas citri. Then potato blight disease, it is Phytophthora infestans. இல்ல அப்பப்போ go through பண்ணிக்கோ, அந்த names ஞாபகம். I hope you follow. So citrus cankerum, potato blight disease. எதுக்கு? Plant diseases. Right? Then microbes in food process. நம்ம வந்து இதுக்கு முன்னாடி வீடியோல detailed super figures ஓட படிச்சொல்லிருக்கோம் இல்லையா? ஒன்னு fermentation, pickling, boiling, sugaring. Fermentation வந்து starch, sugar to alcohol. Pickling வந்து edible antimicrobial liquid. The chemical pickling வந்து vinegar, alcohol and vegetable oil. Whereas fermentation ல வந்து organic acid. Boiling ல என்னது example? Milk and water. Sugaring ல antimicrobial syrup. I hope you understand. முதல்ல go through பண்ணு traditional technique. இந்த வீடியோ 19 தானே, முன்னாடி வீடியோ ரொம்ப நல்லா இருக்கும், நிறைய பிக்சர் சூப்பரா இருக்கும். அது வந்து go through. Just recollect the points that we have done. That I am expecting from you. Can you? Next, modern technique வந்து pasteurization, it is என்ன சொல்ல demonstrated by Louis Pasteur, and then food production is called probiotics. Lactobacillus acidophilus and bifidobacterium, bifida, they are called probiotics. சரியா? Whereas prions, they are proteinaceous infectious particle as neither DNA or RNA. Example, உனக்கு தெரியல ஜேக்கப் குரு எல்லாம் விட்டுரும் கொஞ்சம் கஷ்டம். புரோட்டீன் வந்து க்ரூஸ்வெல்த் and ஜேக்கப் குரு. Virion, virus present outside the cell. Virions are viruses present outside the cell. சரியா? So இது வந்து modern technique. I hope you are able to follow. இது கொஞ்சம் example கொஞ்சம் கஷ்டம். Just ஒரு go through பண்ணிக்க. So that you will understand better. அப்புறம் இந்த example நம்ம textbookல page 21, sorry, நம்ம textbookல different examples கொடுத்திருக்கு. All that you will go through. I hope you follow. Then this is prions and viruses. Structure of prion. Structure of prion and viruses. And the virus the prion and இது வந்து கொஞ்சம் important. You should know because they are helpful in other higher studies க்கு உனக்கு இது தெரிஞ்சிருக்கணும் prions பத்தி. சரியா? They are as prions and virions. They are highly infective and they are highly attacking very cause வந்து ரொம்ப severe. That is prions and virions. சரியா? இதுல பாரு structure of virion, lipid envelope, protein capsid, outer இது கோட் பேரு protein capsid, then nucleic acid, spike in the projection சொன்னேன் இல்லையா இந்த கொரோனா வைரஸ்ல இருக்கு. Then virion associated with the polymerase enzyme. I hope you understand. So this is for the video 20 where we are completing the lesson microorganisms friend and foe. சரியா? I hope you are able to follow this lesson. எல்லாத்துலயும் the what is it? Doubt clearing la புரிஞ்சுக்கோ. That we will clear the doubt. சரியா? And then in the videos la you noted the whiteboard la sometimes the video number வேற மாதிரி இருக்கும் because some videos with the information I merged it. அதனால video number you don't worry, only the content what we are teaching, what is the link given to you, that alone you understand the lesson. I hope you follow. So this is for video number 20 where we have learnt microorganisms friend and foe. I hope you follow. Are you clear? We will continue in the next video session. Have a good day. Have a nice day. Thank you.